All right, today we're going undercover. We're diving deep into the microscopic world to explore the secret lives of bacteria. And trust me, it's a story packed with high-speed evolution, some serious genetic espionage, and it explains how these tiny organisms became the planet's ultimate survivors. So let's follow the clues and see what we can find.
So here's the big question, right? It's something that keeps scientists up at night and definitely worries doctors. How does a seemingly simple little bacterium suddenly become an unstoppable superbug, totally resistant to our best medicines? And how does it happen so incredibly fast? Well, the answer isn't just about slow and steady evolution. It's about a hidden, high-speed network for sharing information.
To crack this case, we have to start by looking at their genetic blueprint. But here's the thing. It's not some rigid single document. It's actually more like a core instruction manual that comes with a whole library of optional plug-and-play expansion packs.
So this blueprint is basically split into two key parts. On one side, you've got the chromosome. Just think of that as the bacterium's core operating system. It has all the absolutely essential genes, the stuff it needs just to live. But then you have these other things called plasmids, and these are the game changers. They're small, separate circles of DNA, kind of like installable apps for your phone. They're not essential for daily life, but they can carry awesome special function genes, like, say, the code for antibiotic resistance. And the most important part? They can be copied and shared.
And if you thought that was cool, it gets even more dynamic. Bacteria also have these things called transposons, which have the awesome nickname jumping genes. And they do exactly what it sounds like. These are little snippets of DNA that can literally cut themselves out of one spot in the genome and paste themselves into a completely different one. They can jump from the main chromosome over to a plasmid, or even from one plasmid to another. It is literally the bacterium's own internal cut-and-paste function for its genetic code.
So get this, even before bacteria start swapping secrets with their neighbors, they have some really powerful ways to generate new traits all on their own. You can think of it as their in-house R&D department, and it runs on two primary tools. The first tool is just plain old mutation, a random typo in the genetic code. Now, a rate of 1 in a million, that sounds like pretty long odds, right? But you have to remember, bacteria deal in absolutely massive numbers. In just a single colony, you've got trillions of cells dividing like crazy. That means at any given moment, there are millions of new mutants just hanging around. And if one of those random typos happens to give it an edge, like resistance to a drug, well, that's the one that's going to survive and thrive.
Their other big internal tool is recombination. Now, this isn't about creating something totally new from scratch. Nope, this is all about the remix. Bacteria can take their existing DNA and just shuffle it around, grabbing bits and pieces to create entirely new genetic combos. It's like a DJ taking two completely different tracks and mashing them up to create a brand new hit.
Okay, now we get to the really juicy part, the main event. Look, mutation and recombination are powerful, but they can be kind of slow. The real secret to how superbugs rise up so fast is what we're calling the genetic spy games. Because bacteria don't just innovate in a vacuum, they actively steal, share, and trade genetic secrets with each other.
And they've got three main techniques for this genetic intelligence gathering. They can literally scavenge DNA from their surroundings, they can use a virus as a secret delivery service, or they can create a direct, secure line to another cell. Each method is its own unique brand of spycraft.
First up, we have transformation. And honestly, this is basically genetic dumpster diving. When a bacterium dies, its cell wall breaks down and all of its genetic material, including those really useful plasmids, just spills out everywhere. A nearby bacterium that's competent can just slurp up these fragments. And if it finds a useful piece of code, like a gene for resistance, it can stitch it right into its own genome. Easy peasy.
Next up is transduction, and this is where things get really clever, almost sneaky. You see, bacteria have their own viruses, and they're called bacteriophages. Sometimes, when one of these viruses is building new copies of itself inside a bacterium, it messes up. It accidentally grabs a piece of the bacterium's DNA instead of its own viral DNA. So when this faulty virus goes and infects a new cell, it's not delivering a virus, it's delivering a genetic gift from the last host. The virus becomes an unwitting microscopic courier.
But the most direct, the most intentional, and probably the most sophisticated method of all is conjugation. This isn't left to chance. It requires direct physical contact between two cells. It's the bacterial equivalent of plugging in a secure cable for a direct file transfer.
So how does this work? Well, it's pretty wild. The donor cell, the one with the goods, grows this long, thin tube called a sex pilus.
It uses it like a grappling hook to latch onto a recipient cell and pull it in close. Then, a bridge forms between them, and voila, the donor cell sends a fresh copy of its plasmid right across. So not only does the recipient get a new skill, but now it also has the plasmid that lets it share with others. The secret agent has just recruited a new agent.
Okay, so spy games, jumping genes, secret handshakes. Why should you care? Because these spy games are precisely how we get superbugs. When we use antibiotics, we put enormous pressure on bacteria, creating a desperate need to survive. And that's when their genetic sharing network kicks into absolute overdrive.
So let's trace the path. Here's how a superbug gets made. It starts with special plasmids, called R plasmids, which happen to carry genes for antibiotic resistance. Then, those jumping genes, the transposons, start hopping onto these plasmids, collecting multiple resistance genes and bundling them into one dangerous package. Finally, conjugation spreads these superpowered R plasmids through the entire bacterial population like wildfire. And just like that, you've turned a whole community of bacteria into superbugs.
But here's the amazing twist in this whole story. For all the danger they pose, these incredibly sophisticated bacterial systems are also a gift, because scientists have figured out how to hijack this ancient genetic machinery for our own benefit. We've learned to hack the hackers. And our playbook? It's basically a refined, souped-up version of what bacteria have been doing for billions of years. We pick a vector, a harmless plasmid or a virus, to carry our gene of interest. We use special things called restriction enzymes as our molecular scissors to make a precise cut. Then we use another enzyme, DNA ligase, as molecular glue to paste our gene right into that vector. Then we just pop that engineered plasmid back into a bacterium and let it do what it does best, make millions and millions of copies.
And the results of this, they've been nothing short of revolutionary. By turning bacteria into these tiny, living factories, we can mass-produce incredibly complex medicines. The insulin that keeps millions of people with diabetes alive, the life-saving growth hormones, powerful antiviral drugs called interferons, even the vaccine for hepatitis B. All of these are modern medical miracles, and they're only possible because we learned to speak the genetic language of bacteria.
So, from creating some of the biggest global health threats to enabling our most life-saving medicines, this bacterial genetic toolkit is one of the most powerful forces on the planet. They literally wrote the original playbook on rapid genetic adaptation millions of years ago. The only real question now is, what will we, using their own tools, write in the next chapter?